Hello everyone, welcome to the course on genetics. Today we will see linkage. It is a new chapter of genetics. In previous videos we have discussed about multiple allelism with its examples. Now we will proceed towards our new chapter, that is linkage. So in this chapter we will discuss about what is linkage, that is definition of linkage, and how linked genes inherited in different types of offsprings. So let us start with the definition of linkage. If the genes are situated in the same chromosome and are fairly close to each other, they tend to be inherited together. So this type of coexistence of two or more genes in the same chromosome is known as linkage. Another definition, it is more simple. When two genes are in the same chromosome, they are tied to one another and are said to be linked. Third definition tells us that tendency of two genes in the same chromosome to remain together in the process of inheritance, it is also known as linkage. So very simple concept is here. If there is a chromosome and if two or more genes are present here, suppose in this case we will suppose there are two types of genes present, and these are present fairly close to each other and they have a coexistence with each other. These two genes are so near to each other and they will inherit together for a particular character or particular phenotypic expression. So according to our second definition, these two genes are present on same chromosomes and they are tied with each other. Means these two genes will be tied with each other and they will not break this linkage, though they are inherited into the offspring also. So such type of two genes which are present on same chromosomes and fairly close to each other and they tend to inherit with each other or inherit together. So these two genes are known as linked genes and the concept is known as linkage. Now we will discuss how these linked genes are inherited. So these linked genes stay together in the same combination as like their parents and don't assort independently. So linked genes will not assort independently. We have selected two parents which are having linked genes. The gene you can see this capital AB, which is present at the left side of the slash. These are present on one chromosome, and the another pair, that is capital AB, it is present on the right side of the slash. They are present on the homologous chromosome. So this is present on a chromosome and these are present on the homologous chromosomes. Here also same, AB and small ab. So these two genes are linked genes and they are present in parents. Suppose we have selected this one parent and this is one parent. They have the linked genes, both they have the linked genes. Now this particular parent, it will produce gamete capital AB. Here also it will produce gamete small ab. So here F1 will be formed. It is again which having linked genes. So capital AB and slash small ab. Now in the second step, we allow these F1 offsprings to cross with the recessive parent, and this is also known as test cross. So we have crossed this resulting progeny with the recessive parent. So they will produce gametes capital A, capital B, small a, small b. So they will produce only two types of gametes because they cannot assort independently as they are linked genes. So they will produce only two gametes, or they will produce two F1 gametes only. this will produce only one gamete, that is AB. Now if we cross them, this capital AB and this small AB, it will forms capital AB, small AB. This will form small ab, small ab. So here you can see that the linked genes will stay together in the same combination as like their parents and don't assort independently. So here you can see this is one parent which was having linked genes. This is offspring which is also having linked genes as like its parents. So they will produce the genes, or they will produce offspring as like their parents. So linked genes, they stay together in their offspring also as like their parents. In this case also you can see this is the first parent in test cross we have used. It is the F1 offspring. We have selected it for next cross, test cross. So now this is a parent, but here you can see the combination of genes or genes are linked, capital A, capital B, small a, small b. These are again similar to, so offspring is similar to parent. So linked genes, as per our statement, linked genes stays together in the same combination as like their parents and don't assort independently. They will not assort independently. So only two types of offspring have been formed, so they will give rise to ratio 1:1. Now the genes, if genes would have not linked, they may be assorted independently and resulted in the ratio 1:1:1:1 instead of 1:1. In the case where genes are linked, we have clearly seen only two types of offspring have been formed and they are similar to their parents. Means as they are linked with each other, they will produce similar types of genes in their offspring also when the genes are linked. But if genes are not linked, they will assort independently. How they will assort independently, we will see. We have selected two parents which are having no linked genes. See here, no slash is shown here. Here also no slash. So genes are not linked. So capital A, capital A, capital B, small a, small b, small b. They are parents we have selected. In this case, gametes, they will give rise to capital AB, small ab. F1 will be produced. It is no doubt, it is hybrid one and it is heterozygous. It will form capital A, small a, capital B, small b. So this is our F1 offspring. So it is very different from linked genes. Now see here, we allow them also to test cross. So here this is our F1 offspring, capital A, small a, capital B, small b, and we allow them to test cross or cross with our recessive parents, small a, small b, small b. Now as the genes are not linked, they will assort independently and they will produce four types of gametes. So here you can see, as per our the rule of independent assortment, capital A, capital B, capital A, small b, small a, capital B, small a, small b. So these are four offsprings formed due to independent assortment. Here you can see only one gamete it is formed, small a, small b, and it will give rise to the F2 offspring. That is four offsprings have been formed. In linked genes only two offspring have been formed, but here four offsprings are formed, and out of them 1/4 are capital A, small a, capital B, small b. 1/4 are capital A, small b, small b. 1/4 offsprings are small a, capital B small b, and 1/4 are small a, small b, small b. So all these 4 offsprings have been formed into the ratio 1:1:1:1:1. So this is called as the independent assortment. So linkage, it is totally different than the independent assortment. There is a difference that in linked genes we can see only 2 offsprings have been formed and they are as like their parents. But in the case of this independent assortment, 4 types of offsprings are formed and they are some are as like their parents and some are intermediate. So see here, these are as like parents, these 2, and remaining 2 they are intermediate to the parents. So this is known as independent assortment, which is clearly different from the linkage. These are some references used. Thank you for your attention.